This is a production of Cornell University.
Okay, good. Yeah, so Shataya mentioned I'm a third-year PhD student in the Larry Smart Lab up at Cornell Agritech. And my research really focuses on looking at the mechanisms and pathways of sex determination in the dioecious bioenergy crop, Salix pururea. So a little bit of background. Salix, the genus Salix contains over 300 species. Almost all of these are dioecious. And shrub willow belongs to the section Vitrix. And these are short rotation coppice bioenergy crops. So they're grown in high density plantings such as this. And they can be coppiced every 2 to 4 years. And then the chips are burned to generate heat, which can also be used to produce electricity. So my research really focuses on the species Salix pururea as a model system for the shrub willows. So here you can see the female catkins or flowers and the male catkins or flowers. And Salix is also a close relative of the genus Populus, the poplars, which are another important biomass and paper crop. Okay, so first we need to talk a little bit more about the heterogametic systems in angiosperms. So only about 5% of angiosperms are dioecious, and these can contain either ZW or XY systems. So the ZW system is the female heterogametic system. And this is where the females are ZW and the males are ZZ. So this is, you're probably most familiar with this as a system that's present in birds. Kiwi fruit are an example of a crop that actually have a ZW system. And then this is in contrast to the XY system, where the males are the heterogametic sex that are XY and the females are XX. So this is the system that's present in mammals, including humans, and some crops such as papaya and asparagus. So, but what's really interesting about Salix as well as poplar is that both these genera have both XY and ZW systems in the same genus. And then in Salix pururea in particular, the SDR, the sex determination region, is on a portion of chromosome 15 that contains about 400 genes. So here is a phylogeny that was generated by some of our collaborators. And so this is of the whole Salicaceae family. So that includes Salix and the sister genus Populus, the poplars. And what's unique about the Salicaceae family, well, one of the many unique things is that they have homomorphic sex chromosomes. So that means that most of the sequence across these chromosomes is conserved, and so you can't really distinguish sex chromosomes by karyotyping or flow cytometry like you would in, say, a lot of animal species. And so the sex determination regions are non-recombining portions of autosomes, essentially. So in Salix pururea, it has a ZW system that's present on chromosome 15, and this takes up about 6.8 megabases, which, as I mentioned earlier, has about 400 genes, and that's about 40% of chromosome 15. So the other 60% of that chromosome is essentially an autosome. So this presents a pretty big challenge when trying to determine master regulators of sex. And so most of the other shrub willows, like Salix pururea, seem to have a chromosome 15 ZW system. But as we get to some of the more basal Salix, such as Salix exigua, while they still have a chromosome 15 SDR, it's actually an XY system. And then in the most basal of the Salix, such as Salix nigra, it's actually chromosome 7 XY system. And then we see in the poplars, chromosome 19XY, chromosome 19ZW, and chromosome 14XY systems as well. And so this is really interesting and indicates that despite dioecia being conserved across the family, the sex determination region has both shifted chromosomes and changed heterogametic systems multiple times throughout the evolution of this family, and really presents Salicaceae and the genus Salix in particular as an excellent resource to study sex determination in angiosperms. So for my research, we made use of RNA-seq data collected from 90 male and 90 female catkins from an F2 family with the help of a JGI. And so this F2 family was generated by crossing our reference male individual and our reference female individuals, for which we have fully sequenced genomes, and then doing sibling mating from two of the F1 individuals, and then getting the F2 family. And this is kind of a summary of the bioinformatic pipeline I ran through, and I won't go into too much detail on this, but if you have more questions, I'd be happy to answer them. But basically, I used DE-seq to do differential expression, WGCNA to do network analysis, a pathway analysis in the cytoscape app Klugo, EQTL mapping using matrix EQTL, and we also had some small RNA-seq data that we were able to analyze. And this MA plot here, the x-axis is the overall expression of the genes, and the y-axis is the log2 fold change in expression. So positive values are more male differentially expressed and negative values are more female differentially expressed. So right away, you can see that there are thousands of genes that are differentially expressed. These are located across the genome. And so sex dimorphism is widespread genomically. But my research really wants to get at what are the master regulator genes in the sex determination region. So existing models of sex determination posit there's likely only one or two genes that control sex determination. And then also I'm interested in what are the intermediate genes between those one or two sex determination genes and these thousands of genes across the genome that are sexually dimorphic. So I use some of these approaches to start to tease that apart and get at those. So using these different methods, I identified a series of candidate genes located in the SDR, so narrowing it down from about 400 genes to about 8 genes. And so these are genes that are, first of all, they're unique to the WSDR, so they're present on chromosome 15W and in females, but absent from 15Z and thus absent from males. In the network analysis I did, they had module membership to highly female correlated modules. And they also had extreme and significant differential expression. And lastly, genes that had functional annotation that is consistent with something that might be involved in sex dimorphism. So first I'll mention these AR17 homologs. So these are, in Arabidopsis anyway, they're a negative regulator of cytokinin. And there's 4 copies here in the SDR. And these are our strongest candidates because they're also present in the poplar SDR on chromosome 19. And cytokinin signaling has been implicated in the sex determination pathways in both dioecious and monoecious crops. So kiwi and persimmon that are dioecious crops. And then also cytokinin signaling is involved in female flower development in the cucurbits. And so this column here is the male to female log2 fold change. So negative values are more female differentially expressed. So all these candidate genes are highly female differentially expressed, which makes sense because they're in they're present only in females. We also had expression data though from an unrelated monoecious line. So a line that has both male and female flowers on the same catkin. And so this shows the monoecious to female and then the monoecious to male differential expression. So for these AR17 genes, you see they're highly expressed in female. They're also have little to no expression in the monoecious line relative to the female and about the same in male, between male and monoecious, which is about nothing. So this indicates that these might be male suppressor genes. There's also an Argonaut 4 homolog and a double-stranded RNA binding 1 protein. And so these are interesting because the terms, so one controls DNA methylation, one controls mRNA silencing, and these are both enriched Go terms in our bulk RNA-seq data set. So these are also strong candidates, and they, like the AR17, also show very low expression in the monoecious in males, which indicates that they are likely male suppressors. There's also this kind of unannotated zinc finger transcription factor that is also maybe a male suppressor. And there's one gene that we think might be a female inducer, so a gene that actually promotes female traits. This is a Gata15 transcription factor. This is involved in floral and meristem development. And it shows very high expression in females relative to males, no significant difference between the monoecious and the females, and very low expression in the males relative to monoecious, and indicates that it might therefore be a female inducer. So these are some of the candidate genes we identified. I also did EQTL analysis. So EQTL analysis looks at the expression of genes and tries to correlate those to polymorphisms across the genome. So doing this EQTL analysis, we identified 54 genes that had significant associations with the SDR that were involved in floral development and secondary metabolism. And these are the 2 primary sex dimorphic phenotypes that we can observe in catkin tissue. So among these were multiple MADSBOX and other floral development genes, as well as numerous phenylpropanoid and terpenoid pathway genes, which are involved in secondary metabolism. We also identified 85 transcription factors that were associated with the sex determination region. Among these transcription factors involved in secondary metabolism, floral development, and gibberellin signaling as well. And so these are candidate genes for being intermediates between the sex determination region and the thousands of genes downstream that are differentially expressed. So all these were determined bioinformatically, but we really need to validate these using functional data. So this is kind of the next step that I hope to accomplish in the next couple of years. We want to do DAP-seq and ChIP-seq to really validate the binding sites of some of those transcription factors. We want to do bisulfite sequencing to look at genome-wide methylation that might implicate that Argonaute 4 in being involved. Validating these candidate genes through functional knockouts and overexpression in models such as poplar and Arabidopsis. We unfortunately can't do this in Willow because it hasn't been successfully transformed. And we'd also like to do protein-protein interaction analyses on some of the other non-transcription factor candidate genes in the SDR to see if they're actually involved in their canonical pathways and what that might mean for sex determination. And with that, I would like to thank all the current and former members of the Larry Smart Lab, as well as our collaborators across the US and China at various universities, and then the NSF grant that has supported this research. And with that, I'll open it up to questions.
There is one question there.
Yeah, okay. The question is, how can breeders use the information about the location of sex determination genes in their breeding program? Yes, that's an excellent question. So for one, you know, to breed Willow, you need a male and a female. So that alone, to be able to select, and I didn't really talk about that, but one thing I am working on is developing markers to actually select for male and female from seedlings. Another really big use is that a lot of the secondary metabolites, as well as some actually biomass traits, are correlated with certain sexes. And so if you're breeding for bioenergy or biomass, knowing what sex a plant is can be very useful. So you can select for sex, which is really easy to phenotype. And then you can get correlated biomass traits along with that.
This has been a production of Cornell University, on the web at cornell.edu.